I am Lakshmi Prasanna, Assistant Professor, Department of ECE, Institute of Aeronautical Engineering. Welcome to the lecture session of image processing. In this session, we will discuss about elements of visual perception. So, if we go internally into this topic, all these are different topics we are going to cover today. So, let us see what are all the topics. So here, first question is, why to learn the structure of human eye? Why it is essential to learn the structure of human eye? And next one, biological aspects of human eye. That means what are different internal and external parts that are available in the human eye and what are their functions. And next one, image formation in the eye. After learning the structure of human eye, we will learn if we see an image, which is any image which is around us, generally how it will be reflected in our eye, how that particular image will be formed in the eye. And next important topic is brightness adoption and discrimination. So all these are different topics we are going to learn in this session. So let us go in detail. First question is, why to learn the structure of human eye? So generally in digital image processing, whatever we see, or generally in digital image processing, it is completely built with the mathematical formulas and probabilistic formulas. And based on this only, digital image processing, the human being will have their own intuition, and also based on the structure of human eye only, the person is going to select any particular technique. And also one important point, in the human being, eye is one of the important sensor. So that is why initially we need to learn the structure of human eye. Now, coming to the biological aspects, in image processing application, images are produced as output. For example, for any system, image will be given as input. After it is processed, again it is going to produce the output as the processed image. It will also be in terms of image only. Somewhat modification will be done on the image. So in image processing applications, images are produced as outputs. So as images are produced as output, there is one important advantage. If we see the output as an image, it is easy for human observations. That is, human being can be easily recognizing that particular objects. Now, coming to the human visual system, as we are learning about the structure of human eye, it basically consists of three important parts. The three important parts are, one is eye, optic nerve, and another one is brain. If you want to relate this to the digital image processing, this eye can be related with image sensor, or eye can be related with the camera. And another important visual system, it is optic nerve. This optic nerve can be related with the transmission path. And coming to brain, this brain can be related with image information processing unit, or the brain can be related with computer. So these are the three important parts in human visual system. Now, so in human, the most important sensor is eye. So we need to learn about the structure of eye. So here you can see this figure which shows the structure of human eye. It consists of different parts like eyelid, pupil, sclera and iris. We will be discussing about all these parts in detail while learning about the internal and external parts of the human eye. If you observe this structure of human eye, it is spherical in shape and it has the average diameter of approximately 20 millimeters. So coming to the structure of human eye, it is classified into two categories. One is external structure of eye and another one is internal structure of eye. So in the external structure of eye, there are different parts. We can see here sclera, conjunctiva, cornea, iris and pupil. All these are different parts in the external structure of eye. We will be discussing about in detail about all these parts and what is the function of each one. And coming to the internal structure of human eye, it consists of different parts like lens, retina, optic nerve, aqueous humor and vitreous humor. And all these parts and its functions we are going to discuss now. So totally these are the external and internal parts of the human eye. Coming to the detailed discussion about all these parts and the function, we need to learn about cross section of human eye. So this is the cross sectional structure of human eye. So whatever the internal and external parts we have discussed in the previous slide, all these parts can be observed in this figure. So we can see the cross sectional of human eye. It consists of different parts like ciliary muscle, cornea, iris, lens, aqueous humor, sclera and medial rectus muscle and vitreous humor, optic nerve, macula, choroid, retina and lateral rectus muscle. So all these are different internal and external parts we can observe in this cross section of human eye. So if we observe this cross section of human eye, basically it consists of mainly three layers. So here the three layers are, first one is cornea. So we can observe here, this is cornea. We can see this is the external membrane of this human eye. That is the external layer of the human eye. And next one is sclera. So here this part represents sclera. That means cornea is the external membrane. After cornea, the next internal membrane will be sclera. Layer will be sclera. And the third one is choroid. Under sclera, you will have another layer which is called choroid. These are the three layers in this cross section of human eye. So external most layer will be cornea. Next layer under that cornea you will have sclera. Under sclera we will have a choroid layer. Now we will discuss about all these internal and external parts of this human eye in detail. Now, so as we have seen, it consists of basically three layers. The three layers are cornea, sclera and choroid. So if you observe this cross section of the human eye, we can see here cornea is available and another one sclera and the next one is choroid. Now we will see what are their functions. So if we see here, cornea is the first external layer. So it is a tough transparent tissue that covers the anterior surface of the eye. That is the external most layer. And next important part layer is sclera. So under cornea, we will be having the next second layer, that is sclera. Sclera is an optic membrane that encloses the remainder of the optic globe. And after sclera, we will be having the third layer, which is called choroid. So choroid is the layer which will be lying under sclera layer. So what this choroid layer consists of means, this layer will consist of a network of blood vessels. So through these blood vessels only, sufficient amount of nutrition will be provided to the eye. So these are the important three layers in the cross section of human eye. Coming to the next parts. Next one is iris. So iris can be considered as the eyeball. Okay. So here we can see the iris. Here we can see the iris. Iris can be considered as an eyeball. And here in the eye, most visual part is iris only. And here the central portion of the front of the eyeball is termed as iris. Iris is a pigmented structure. Generally, all the human beings will not have eye color same. Some of their eye color may be black, some of their eye color may be brown, and some of them will be having blue eyes. Okay. So all these color represents the color of iris. So it is defined by the pigmentation of iris. So here this eyeball is called iris, but in the center of this eyeball, we will be having one black color hole located, and that part generally is called as pupil. It will be located at the center of iris. That is called as pupil. Now, coming to the next part, which is called pupil. So here, the black color dot or the black color hole that is present in between this iris eyeball is called as pupil. Okay. So generally if we see iris, we can see hole, black color hole. That hole will be at the center of the iris. It is called pupil. So it is a small aperture located in the center of iris, and its diameter will be approximately 2 to 8 millimeters. So it allows light to enter and focus on the retina. So generally the light will be entering from this pupil, that is small hole, black hole, which is present in the center of the eye, and through that pupil only it will be falling on the retina. And coming to the next part, aqueous humor. So here we can see this is aqueous humor. So this aqueous humor will be present here. Okay. So based on this cross section of the human eye, this aqueous humor is a watery like substance. It will be in the form of liquid, and this liquid generally it will be available in between two parts. What are the two parts means? It will be available between cornea and lens. So you can see here, this is lens, here it will be, and we can see here cornea. Okay. So this aqueous humor will be present in between this cornea and lens, and it will be water-like substance. It will be in the liquid form. So what is the purpose of that means? It will nourish the eye and keeps it inflated. Now, next part in the cross-sectional of the human eye is lens. So if you see the cross-sectional of this human eye, this part is called lens. So here this part is called lens. Okay. So the lens is made up of concentric layers of fibrous cells and it is suspended by a fiber that attached to the ciliary body. Okay. So this lens will be attached with the help of this ciliary muscle. So generally it consists of 60 to 70% of water and 6% of fat and more proteins that will be provided to the tissues in the eye. So generally what is the main function of this lens means? So the lens will be colored by a slightly yellow. That means, for example, if we consider the age of a person, as the age of the person increases, the lens color will be changing. Slowly it will be becoming yellow and the vision also will be getting degraded. So it will be based on the lens. So the lens will be colored by slightly yellow pigmentation as the age increases. So in extreme cases, in different cases, it leads to poor color discrimination and loss of color vision. So if this becomes totally yellow, then that particular person will lose his vision. So the lens absorbed approximately 8% of visible light spectrum. So the light will be absorbed by this lens through pupil and it will be falling on the important part which is called retina. If that falls on the retina only, that particular object can be visualized by a human. And that is what the next important part is retina. So it is the innermost membrane of the eye. So here we can observe retina. It is the innermost layer of the eye. Light from any object outside the eye will be falling on the retina and that light after being getting refracted, the image can be visualized by that particular person. So we will discuss in detail about this retina. First, light will enter through pupil and lens and then it falls on the retina and inside that retina we will be having different set of nerves and all that nerves will be connected to one important nerve which is called optic nerve. Through that optic nerve it will be, the information will be going to the brain and the brain is going to identify the object. So we are going to discuss in detail about this retina. Now, next important part in this cross-sectional diagram of human eye is vitreous humor. So we can observe here, this is the vitreous humor. It is present in between. Generally, if you look, it will be glassy in structure, but generally it will not be glassy. According to the biological aspects, it will not be glassy. It will be in the form of jelly. So if you observe the space between the lens and retina, the space between the lens and retina is called as vitreous humor. So you can see it is present in between this lens and retina. So the space between this lens and retina is called as vitreous humor. The distance between that lens and retina will be varying from 14 to 17 millimeters. So how this vitreous humor will be means, it will be transparent, it will be like glassy in the form, but to see the substance will be like jelly and it is present in between lens and retina. So the main function of this vitreous humor is to protect the eyes and to maintain the structure. That is, eye will be spherical in shape. So to maintain that spherical shape, this vitreous humor will be helpful. Now, as I told, we will discuss in detail about this retina. Okay, so we can go in detail about this part, retina. So light sensitive cells, that means as soon as the light enters from iris through lens and if it falls on the retina, what happens? That particular light will be in the form of electrical, and that particular electrical signals will be carried from retina to brain. How it will be carried from retina to brain means, on the retina we will be having more number of nerves. So these bunch of nerves will be connected to one single nerve, which is called optic nerve. So this optic nerve will be connected to brain. This optic nerve will be connected to brain. So that particular pattern will be viewed based on the distance of discrete light receptors over the surface of retina. Then the brain is going to visualize what is the pattern and what is the object. Now, coming to here, receptors, we need to discuss in detail about the receptors. So here, receptors means all that nerves that are present on the retina are going to receive the light that is coming to the retina, and then that particular receptors are going to send to optic nerve, then to brain. So here, coming to receptors, receptors are classified into two classes. So here the two classes are cones and rods. So here, what the cones consist of, what are the features the cones will be having. So number of cones that will be available are 6 to 7 million, and that cones will be present at the central portion of retina. So here you can see retina. Okay, if the light falls onto the retina, it should be falling on the central part of retina. Then only it is capable of identifying that particular image. And that central part of retina is called as fova. And cones are highly sensitive to color. So based on this number of cones only, we are going to identify all the colors in the image. Humans can resolve fine details, and cones consist of number of nerves. Here each nerve is connected to its own nerve. Each cone will be connected to the own nerve. And here cone vision is called photopic or else bright light vision. We will see some example in order to identify clearly what is cone and what is rod. Coming to rods, number of rods available are 75 to 150 million, and rods are connected to a single nerve end, and these rods are not involved in color vision, and rods are sensitive to low level of illumination. So in order to clearly understand this cones and rods, we can take one example. So we can see the object, or we can see any image in bright light. So when it is seen, cones will be activated in our eye. As cones are activated on the image, particularly all the colors can be visualized. If the same image is viewed in the darkness, at that time this rods will be activated. Hence, during the darkness, we are not going to identify the colors. We are not going to visualize colors on that image. So here, cones will be activated when the light is bright. So we can see all the colors in the image. Rods will be activated during when the light is dull or when there is no light. So the colors on the image cannot be visualized. So that is the main difference between cones and rods, which comes under receptors. So here, as we have discussed, cones are active at higher light levels. That means when the light is available. Hence it is called photopic vision. So it is capable of color vision. Human beings can see the color vision. Whereas rods are responsible for vision at low light levels. So during dark room and all, these rods will be activated. So such type of vision is called scotopic vision. So as we have discussed the example, objects that appear bright colored in daylight, if the same objects are seen during the moonlight, they will appear colorless because only rods will be activated at that time. So this is the main difference between the two classes of receptors, cones and rods. Now, so in order to clearly know at what time the cones will be activated, at what time the rods will be activated. So this is the graph that will clearly say the difference. So here it shows the distribution of rods and cones in the retina. So here is a simple graph, degree from visual axis versus number of rods or cones per millimeter square. So if you observe here 0 degrees, before that, if you see here the continuous lines like this, here the continuous line is available. Okay, so this continuous line we can see here. Okay, so this continuous line are called cones, whereas here we can see the dotted lines are available. The dotted lines are called rods. So whenever light falls on the retina, especially it should fall at the center of retina. So here when the degree is at 0, here we will have high intensity for cones. Here we will have high intensity for cones. So as the other than that 0 degree center, if you see here rods, the intensity is decreasing and increasing. That means it is not constant. So if you see this part, in this region, no cone is available or no rods are available. So that particular area we will call as blind spot. Nothing can be seen. Either cones are not activated and rods are not activated. Now, so what this graph indicates, it shows rods and cones. So the graph illustrates that cones are present at low density throughout the retina. Okay, so here all the density of the cones are very low throughout the retina, but whereas it is sharp here. Here it is sharp. Remaining areas it will be low. At the center of the retina, the intensity will be very high. The center of the retina is called as fova. Now, coming to rods. Rods are present at high density throughout the retina. Here you can observe. Throughout the retina, the density of rods are very high. Whereas if you come to the center of the axis, the density of these rods are less. So if you enter the center of the fova, the intensity is declined. So this is the difference between cones and rods in the retina. Now, so that is what about the structure of human eye. Coming to the next topic, that is image formation in the eye. So if you see any object, how that particular object can be formed in the eye? What about the size of that particular object which is formed in our eye? So we are going to clearly discuss here. Now, so this is the figure, graphical representation. So here in this figure, we can see here this is the eye of a person. So here we can see center. So that dot represents center. So it is the center of the lens. So this particular person is watching this palm tree, and the palm tree height is around 15 meters, and from the center of the lens, the palm tree is located 100 meters away, which is too far. Here, from the center of the lens, and here we will have retina. The distance between the center of the lens and the retina is given as 70 millimeters. So considering all these aspects, we have to discuss how the image will be formed in the eye. So here, the distance between the center of the lens and the retina we call as focal length. That means here this 17 mm is given as focal length, and it varies approximately around 14 millimeters to about 17 millimeters. Okay. So here the object is palm tree and it is far away from the center of the eye. It is about 100 meters. When the eye focuses on the object which is far away than 3 meters, then the lens exhibits the lowest refractive power. That means that particular object cannot be visualized properly. From the eye, if the object distance is more than 3 meters, for example, here it is 100 meters, which is far away compared to 3 meters. Such objects cannot be visualized properly as it will be having lowest refractive power. In another case, when the eye focuses on a nearby object, the lens is most strongly refractive. That objects can be visualized very clearly. So based on the distance from the center of the lens and the object that is present, the visualization can be calculated. The distance can be calculated. The height can be calculated. So considering this example with respect to this distance, height and the diameter, here is a simple example in order to calculate the size of the retinal image. So this is the size of the retinal image. In order to calculate what is the size of the retinal image, that is the image formed on the retina of any object what we are seeing. Here we can see a simple example. So to calculate the size of the retinal image of any object, here as we have discussed, the observer is looking at a palm tree where its height is 15 meters and it is at a distance of 100 meters. So here H is measured in millimeter and that should be calculated. So the object of the retinal image can be calculated as 15 by 100. So 15 by 100 is height of the palm tree and distance from the center of the lens to the palm tree. So 15 by 100 is equal to H by 17. So 17 here represents the diameter. So in order to calculate the value of H is equal to 2.55 millimeters. So what is this 2.55 millimeters? This is 2.55 millimeters, the size of the retinal image of that particular object. So this is how the image will be formed in the eye, and that will be the size of the image which is formed in the eye. Now, so coming to the brightness, adoption and discrimination. Here we have discussed already about this rods and cones. Cones will be activated at the bright level, and rods will be activated during darkness. So rods are called scotopic vision and cones are called photopic vision. So if we observe this particular graph, here we can see scotopic and photopic are represented. This is the simple graph which shows the intensity versus subjective brightness. Now, if you consider this graph here, if any person enters into a bright room, that particular person vision in order to see any object, it will be different. And if the same person, if he enter into a dark room, in order to see that object, his perception will be different. But both cannot be done simultaneously. So if person enter into the dark room, his perception will be somewhere here at this particular point. So which is coming under scotopic, because at that case the rods will be activated. If the same person suddenly if he enter into the bright area where the light is high, then his perception will be different. At that time, for example, if we consider here that point, at that time the intensity will be high, cones will be activated. So if we consider this curve comes under photopic, so here we will have that perception, visualization, but both cannot be similar and both cannot be done simultaneously. So that is why here the reason, so the person can visualize, cannot visualize both the things simultaneously. So that is why here it is not possible to cover the entire range from minimum to maximum. So that is why here we are going to consider somewhere at this point where the thing need to be managed. So here the two parts here what I have discussed now. So this point represents a wide range of intensity levels to which human visual system can adopt from stochastic to glare the limit on the order of 10 power 10. Whereas here the common part we have considered where the thing need to be managed. It represents the range of subjective brightness that the eye can perceive when adapted to a level, when adapted to some level. So here the level may be either BA or BB, one level. So in this session we have discussed about the structure of human eye and how the image formation will be there in the eye, and also we have seen the brightness and discrimination, and the content what we have taken is from Digital Image Processing textbook which is written by Gonzalez, and also in order to know the structure of human eye in detail we have referred this particular website. Thank you.
Like, share and subscribe. Hit the bell icon for more updates.